Hello everyone, my name is Sofia Barmak and I am a member of the Behavior Biology Laboratory of the Instituto of Experimental Biology and Medicine. Spatial cognition is a very relevant skill because it involves the ability to orient and navigate in the space, becoming essential because you need this ability to obtain the necessary resources to survive. This skill is present in a lot of zoological species, suggesting it is highly conserved throughout evolution. Our laboratory uses an amphibian model which has a brain structure known as the medial pallium, homologous to the mammalian hippocampal formation. This allows us to study the neural basis and the evolution of this behavior without having a strong cortical modulation. In this study, partially dehydrated male Rhinella arenarum toads were used to learn to find a reward, in this case water, using the egocentric turn as an orientation strategy. A plus maze was used, surrounded by curtains that prevent the outside view. The first 2 days were per training, which consisted of 15 minutes of free time that they were able to explore the maze and obtaining water in both pools. In this picture, you can see how the plus maze looked like. For training, the subjects were separated in 3 groups: right turn, left turn, and control. The control group has a random pool in each session. The training consisted of 3 daily sessions. In each one, the toads had 3 minutes to find the correct pool. If they were correct, they had 2 minutes of reinforcement, but if they chose the wrong pool, they had 1 minute of penalty locked on the wrong side. Then, they were guided to the correct pool, where they had 2 minutes of reinforcement too. The learning criteria was established at 75% correct answers for 3 consecutive days. Once the learning criteria was reached, the brains were extracted for histological processing using the Eisenhower technique, which is an indicator of neuronal activity. The number of nucleolus and its relative size to the nucleus, used as an activity indicator, was determined in the medial pallium region. The results show that the experimental groups, right and left, learned the task and the control group was closer than the random criteria. For histological analysis, Arcturus XT software of the Nikon TE2000 laser dissection microscope was used. Here you can see a picture of a coronal section stained with the Eisenhower technique. In this other picture, you can see what the tissue looks like under the microscope. The nucleolus are seen as the darkest spots, while the nucleus are stained light brown. In the graph, you can see the size of the nucleolus in relation to the nucleus expressed in pixels. The analysis of neural activation revealed the existence of greater relative size of the nucleolus with respect to the nucleus in the medial pallium neurons of experimental subjects. To finish, the behavior results show the ability to the subjects to learn to find the reward using the egocentric turn as a strategy. And these first histological results showed a greater relative size of the nucleolus to the nucleus in the medial pallium neurons of the experimental animals who learned the spatial orientation task. This was not observed in the control subjects. This observed activation reveals that the medial pallium is involved in this learning. In the future, the neuronal activation of other areas of the brain will be analyzed, such as the septum, amygdala, striatum, and the lateral pallium, which could be involved in this learning process. These are the references. Thank you.